Hi all, it's Dr. Clark here, General Biology Lecture Series. Today we're going to discuss chemistry, and we're going to discuss the basics of chemistry. So again, this is a biology class, and as you're expected, um, depending on the career that you're choosing, if you're going pre-med or pre-vet, pre-dent, or wildlife or anything like that, you'll have plenty of chemistry, okay, as you progress. If you're going into nursing and depending on what field you might be going into, this might be the only chemistry that you get. Okay, so it's going to be a brief overview, but there are some pieces of chemistry that biology, well, all of chemistry, biology is directly related to. Okay? But there are pieces of chemistry and some techniques in chemistry that we'll talk about, and some ideas in chemistry like ions and isotopes that pertain directly to the rest of the course. Okay? When we talk about how things move across the membrane and things like that, it's chemistry that we need to know about. Okay? So before I get into the atoms and the ions and the isotopes, I want to talk a little bit about the woman that's pictured here. This is Madame Curie, or Marie Curie. And I'll bring her back up multiple times, but without Madame Curie's research in isotopes, the field of chemistry would probably be still in the dark ages. Okay? She literally dedicated her life to physics and chemistry, and when I say dedicated, she died for her research. Okay, because we didn't know the effects of uranium and on the body. She probably most likely died of cancer due to overexposure to uranium. But nonetheless, I just want to let you know that uh, she's one of my heroes for the research that she's done, and I'm not even a chemist, but because of her research, we've really made breakthroughs in biology, breakthroughs in chemistry, breakthroughs in physics, okay? And it was really due to her foresight and just her ability to go past all of the male versus females issues, and stick her neck out there and let everyone know that just because she's a woman doesn't mean she can't be a scientist. Okay, which really brings me to two books. Okay, the Obsessive Genius, which is about Marie Curie and her work and her research and her life, and is an excellent book. I can't recommend it enough. Okay, and then Women in Science, 50 Fearless Pioneers Who Changed the World. Uh, we don't talk enough about women in science, mainly because it's still a man dominated field. Um, but there was a lot, and has been, and there is currently a lot of females in science that had extreme breakthroughs in science, and much of the material that they did, they lost to their male counterparts. And so I highly recommend this book also. It's an excellent book about the women that kind of paved the way, and a lot of times were forgotten because the male in the room was louder and male. So anyways, pick these books up if you're interested in some excellent reading. Otherwise, I'll talk about a lot of the things that come up in these books, a lot of the people like Elizabeth Blackburn and some other individuals that were really crucial to the field of biology. All right, atoms. Okay, so let's start off talking about the smallest known entity. Okay, and we know this. Remember, we're in biology class, so I'm going to try to tie everything back to why does it matter for biology. We know that organisms are chemical machines. Right? We know that organisms are just built of atoms. Right? And those atoms are linked together to make molecules, and those molecules are linked together to make macromolecules, and the macromolecules linked together to make organelles, and on and on and on. But the basic unit is an atom. Okay? And so I always tell students this: you must know chemistry in order to understand biology. Okay? At least some chemistry. The opposite is not true. To know chemistry, you actually don't need biology. Okay? Now, it will help, okay? But in order to understand biology, you have to have chemistry. And that's why, if you look at a biology degree, that's why biology students are required to take a full year of general chemistry and a full year of organic chemistry at most institutions. Okay? Because chemistry is really the base, okay, the ground level that you have to start with to understand how living organisms interact, how living organisms go about life. Okay? So from that, we can talk about other things like the physics component or chemistry component also, in that anything that is comprised or has mass and occupies space is comprised of matter. Now, this definition may change, not sure, but in the last couple of years, we have been able to, I shouldn't say we, scientists have been able to, physicists have been able to take and make solid light. Okay? Now, light prior to that, really, photons neither had mass nor occupy, well, in some cases they could occupy space, but they weren't considered matter. Okay? And now, since they have a mass, okay, or can be converted to have a mass, this might change kind of the definition of what light is. Okay? But again, that's like we've been talking about before. It doesn't change really much. It changes how we define something, but it doesn't change whether it exists or not. It's still there. Okay? And that might change this definition. All matter is made up of atoms. And we know photons are not made up of atoms. Okay? But if they have mass and they occupy space, then they have matter. So some of these definitions might change, but regardless, I'm just bringing you up to date on new discoveries within kind of the last few years. So all matter is made of atoms. Okay, so what is an atom? Okay, all atoms have the same basic structure. Okay, pretty simple. They have a nucleus, okay, which is comprised of protons, which have a positive charge, and neutrons, which have no associated charge or are often considered neutral. Okay, now, just to make sure that you understand, we will use the term nucleus to describe atoms, or the center or the core of an atom. We'll use nucleus to also describe the center or the core of a cell. Now, these two things are very different from each other. Okay? But the term nucleus really means center or core. Okay? And it gets interchangeably used between chemistry and biology. So just make sure that when you're looking at things, that if we're talking about the nucleus of an atom, it's protons and neutrons. If we're talking about the nucleus of a cell, it's normally the nucleolus, the DNA, ribosomes, these kind of structures. Okay, and we'll get to those in a little bit. Okay, so around this nucleus, this atom's nucleus, is a cloud of subatomic particles, which we call electrons, and they have a negative charge. Okay, and this is very important. Okay, I'm not trying to take anything away from protons and neutrons, But when it comes to biology, the electrons are probably the most important piece, because electrons are what give off energy. Okay? And so when we talk about energy from a biology standpoint, we're often talking about the electrons, either free electrons or an electron transport system, okay, where electrons have been broken free from the protons and neutrons and can be used to drive work. Okay? Now, when we look at atoms in general, okay, we can talk about atoms on two kind of fronts. We can talk about their atomic number, which is how many protons they have in the nucleus. Okay? And any time you're talking about a certain number of protons, you're talking about the same element. So if we're talking about a proton number of 6, we're talking about carbon. Okay? Regardless of the other players, regardless of how many electrons, regardless of how many neutrons, if we're talking about a proton number of 6, we're talking about the element carbon. Okay? You change the proton number, you change the element. Okay? And then we can talk about the mass number. Okay? The mass number is the number of protons plus the number of neutrons. Okay? And so it's basically how much mass does the nucleus have? Because electrons really, they don't have much mass. Okay? And we're interested in kind of the core or the nucleus mass. Okay? The mass number is also important, okay, but does not dictate the element. So you can change the mass number without changing the element, okay, as long as you're not changing the number of protons. So in other words, you can change the number of neutrons, okay, and change the mass number with still maintaining the same element. And we'll come back to what I'm talking about there. Okay? So why I said again and reiterate that electrons are probably the most important piece that we talk about in biology is because the electrons are energy, okay? And the electrons also determine the behavior of the atom. So the electrons, when we talk about them and we talk about them in their orbitals or around the nucleus, okay, their interaction with the nucleus or their interaction or the distance they are from the nucleus will dictate how they interact with nature or how they interact with another atom. Okay? And so electrons are associated with energy, okay? And their energy is based on their position around the nucleus, okay? And this is called the potential energy. Okay? Now around the atom, okay, you will have electron shells. These are places where the electron resides, okay? And it has orbitals, okay, in which are the basic volume or space inside these electron shells, okay, where the electrons are most likely to be found. Now, it's not always that the electron will be in that given space, but they do have a kind of a home in which they live, okay, and they orbit around the nucleus, hence the word orbital. Okay? Now, basic chemistry. Okay? Each electron shell has a specific number of orbitals, and that can be filled by electrons. Okay? If a orbital is incomplete and doesn't have enough electrons, it's often more reactive than a complete orbital. Okay? And so if you have an incomplete orbital, and so it needs electrons, okay, it's either more likely to lose an electron, and this depends on electron negativity, okay, gain an electron, or sometimes share electrons with a neighboring atom. Okay? And so I shouldn't say the goal, it's not like they're consciously thinking, but the overarching premise is that electrons will fill these orbitals. Okay? And when they fill these orbitals, they become more stable. Okay? And so by sharing, losing, gaining electrons, they can complete or fill their outermost electron shell. And this is really the premise behind what we call chemical bonds, okay, or bonds between two atoms. They're sharing electrons, okay, between those two nuclei of those atoms. Okay? Or, you can have bonds between lots of things, but if we're just talking about two, that's what we're talking about. Okay? So back to the potential energy of an electron. As an electron moves to a lower energy level or closer to the nucleus, energy is released. Okay? So as you move an electron from outer orbitals closer to the nucleus, energy is released. Okay? As you pull electrons away from the nucleus and you move them out, okay, to outside orbitals, energy is required. So you can think of it just like a magnet, okay? If you're moving an electron towards a negative charge, towards a positive charge, eventually they'll snap together. Okay? But trying to get that electron, that negative charge, away from the positive, you're going to have to apply some energy. You're going to have to pull it apart. You're going to have to pull it away. Okay? And we'll get to these points in a little bit. Okay? So that sort of brings me to ions and isotopes. Okay? An ion is when an atom has gained or lost one or more electrons. Okay? It can happen in many ways. You can have ions because molecules have been broken apart. You can have ions because an electron has been stolen from another atom. Okay? There's lots of ways at which an ion can occur. An isotope, on the other hand, is when the nucleus of an atom has the same number of protons, but you have an increase or decrease in the number of neutrons. Okay? So the element itself is the same, okay, but its behavior differs often. Okay? And so when we look at elements on a periodic table, so if you remember the periodic table and you're looking at all the elements, okay, they all have a set number of protons, okay, but they don't necessarily have a set number of neutrons. And in nature, we actually have elements that have lots of different mixtures of number of neutrons, or lots of different isotopes. Okay? For example, so if we look at carbon-12, this is the normal, what we call the normal isotope of carbon, okay, which means it has 6 protons and 6 neutrons. Add them together, and that's where the 12 comes. Okay? This is kind of normal, and this is what we'd expect in nature. Okay? And 99% of all carbon that we encounter is of this form, carbon-12. Okay? So 6 protons, 6 neutrons, 6 electrons. Okay? But there's carbon-13 and carbon-14, okay, which means that they have a different number of neutrons, but it's still the same number of protons. Okay? And in this case, you can see the electrons didn't change either. But the electrons can change, and it still doesn't change that it's carbon. Okay? Or they have the potential to change, and it still doesn't change that it's carbon. Okay? But why does this matter? Well, to biology, this was a huge discovery. Okay? And this is one of Madame Curie's major discoveries, is that we have these isotopic forms of each element. Okay? And the big piece that she discovered was that they break up, right, at different rates. Okay? And for that matter, not only do they break up at different rates, they decay at known rates. Okay? So carbon-13 is going to decay at a different rate than carbon-14 and carbon-12. And this is what we call radioactive decay. If you know that rate, then you can start looking at the ratio of, say, carbon-12 to carbon-14 in a piece of material. And as the ratio differs, that can give you an idea of, well, if you know how fast the half-life of an isotope is, okay, then you would know how long that organism has been dead, or how long that rock has been sitting on, sitting on the surface per se. Okay, how long has that rock been, no longer part of the mantle? Okay? And so, apart from just dating fossils and rocks and things like that, radioactive isotopes has also given us the ability to detect cancer or tumors, or in some cases breaks in bone, and torn ligaments and all kinds of things, because if you put a known radioactive isotope onto something like maybe a sugar, like glucose, right, and then you inject it into the body of an individual, that isotope is going to go to a region that needs glucose the most. Okay? And those are regions of which might be growing, okay, extremely fast. And so if you get this isotope into the system and it flows to an area where there's fast cell growth, okay, if we use a marker that can glow, or that we can pick up the radioactive properties, then we can see areas of fast growth. Okay? And that is what we've been doing for a long period of time, using radioactive tracers to identify cancer, or identify bone fragments and breaks in bones and things like that. When you get a bone scan, it's a similar situation. So it's not a system that is very damaging to individuals. I'm not going to say there's no damage or no potential for damage, but the potential is very, very small, because the isotope that's added has a very, very short life. Right? And so some of you have maybe had this done before. You will take whatever the chemical is, whatever the isotope is, either by vein, or sometimes you can drink it. Okay? And then they'll tell you, okay, we need to wait a certain period of time, hour, 2 hours, whatever, and then you have to have the scan done. Okay? So they're waiting for the isotope to flow throughout the entire body, okay, and go to regions where there's active growth, okay, and then you can scan it and see what's happening. Okay? And so these are what we call tracers. Not only can we trace things like cancer, but we're now capable of using tracers to look at biochemical pathways. So even more minute systems, like where in the body is this hormone made, where in the body are these receptors. Okay? And so tracers are now used a lot more often to figure out, well, what location, okay, is this drug what location does this drug potentially have an effect on? Okay, so if you're taking, a like morphine or something like that, where is it going to be, absorbed? Where are the receptors that would pick that drug up? Okay? And in some cases, there might be a lot more in the head region versus the torso, or even the feet. Okay? And for medical purposes, we need to know those kind of things. Okay? I have a little video here. I highly suggest that you click on it and watch it. It really talks about these radioactive isotope tracers and modern medicine. It's an excellent video that shows, kind of in a cartoon form in many cases of where the isotope's going, what it means to have these, and the breakthrough that this means for things like detection of cancer and bone fragments and all kinds of things for that matter. Um, really important research done with this, and there's lots more research to go. Okay, we now get the capabilities of tracking very minute um, atoms, very minute molecules throughout the system to look at, okay, are they getting deposited somewhere? Is there a storage vesicle? How are they getting into cells? These kind of things. And that's just more of that is to come, okay? To the point where we can now trace even DNA, potentially like large strands of DNA or chromosome movement and things like that. With these kind of tracers. So lots of cool things going on. Okay? But from my standpoint, okay, the best comes with dating fossils. Okay? As an evolutionary ecologist, the ability to date something, okay, has drastically changed how we look at the world. Okay? And so knowing the rate of radioactive decay and knowing that it's constant for a lot of elements, we now have the ability to not only measure when that organism was around, but date it with very good confidence. Right? And date it within, kind of 1,000 years in many cases, right? Which will give us the ability to say, okay, no, humans weren't in North America 13,000 years ago, but rather they were all the way to Colorado 15,000 years ago. Okay? Now, this kind of information allows us to make predictions and allows us to realize, well, if man's been in Colorado, thus they've also been in Wyoming, for 15,000 years, okay, then they must have had interactions with this group of organisms or this plant type, okay, that were around during that period of time. Okay? That's important from an evolutionary ecologist's point of view. And the way the isotopes work is, you have these half-lives or rates of decay. The older the fossil is, the greater the fraction of decay, and to a point, and then it's not detectable anymore. Okay? So let's look at kind of an example of this. Okay? So let's say, you have cosmic rays, you have photons coming from the sun, okay? And plants are absorbing that. They're absorbing atmospheric carbon dioxide, okay, to do photosynthesis, and then they're building carbohydrates from that. Okay? So carbon is the base of this carbohydrate. A rabbit goes over, consumes plant material, okay, and takes in carbon-14 and carbon-12, okay, just like the plant did. Okay? Eventually that rabbit dies. Okay? When it dies, no new carbon-14 or carbon-12 is added to the body. So if we know the half-life of carbon-14, for example, is 5,730 years, okay, then we can take and we can look at what's the ratio, what's left in the skeleton of this organism, what's the ratio of carbon-14 to carbon-12. Okay? And as that progresses and that gets less and less and less, then we know the age of that organism is older and older and older. Now, a lot of people have asked me, well, if the half-life of carbon-14 is 5,730 years, okay, somewhere in there, then how do we date dinosaurs? Okay? Dinosaurs are, millions of years old, okay? 300 million years old. Okay? How are you dating dinosaurs? There's not anything. There's no way that you could date it with carbon-14. And they're right. We don't date dinosaurs with carbon-14. Carbon-14 is about up to about 30,000 years. about the history of humans is about as good as carbon-14, humans, Homo sapiens, okay, is about as good as carbon-14 is going to do. Okay? But what we do use is other isotopes. Other isotopes like potassium-40, okay, which has a much greater half-life, a billion-year half-life. Okay? We can use that, and potassium-40 is going to decay at a known rate. Okay? And when I say decay, what I mean is it's not that carbon-14 is going away. The carbon-14 is changing into nitrogen-14. Okay? And so what's happening is the carbon-14 is kind of going away, but nitrogen is in the presence of it. And it's just like potassium. It's called their sister molecule. Okay? So carbon-14's sister molecule is nitrogen-14. Okay? It's potassium-40, which has, a billion-year half-life. Okay? Its sister molecule is argon-40. Okay? They have the same mass, okay? And it's just that ratio of protons to neutrons is changing, okay? And so when you think about radioactive isotope dating, okay, I don't want people to think, oh, well, carbon-14 can't be used on dinosaurs, and people don't actually know how old dinosaurs are. We know how old dinosaurs are, and they're exactly right. Carbon-14 is not used on dinosaurs. Its half-life is too short. Okay? Carbon-14 is used on human things, okay, since humanity's been around. Things like potassium and uranium, and there's lots of other elements that have half-lives that are much greater than a 5,730 year half-life. And that's what's used to date dinosaurs. Okay. All right, so with that, we'll keep talking about chemistry. I'm going to talk about molecules next. So atoms linking together, talking about chemical bonds and things like that. So stay tuned.